So the title contains a question mark, which is very important. Sir asked me, question mark रखना है क्या? Yes, so genetics and cancer. So it means that for study to know the cause to cure cancer. Is it possible? Let's see. It's not easy to say that cure actually is not the word in cancer. What they say is remission, disease free survival, and all that. And here, cancer is, as you know, in any book you open for cancer, a textbook, it will start with it's a multifactorial, multi step, complex disease. So, which uh, explains why there are so many cancer research centers and why there is so much of cancer research going on. Whereas other diseases, where the cause is known, it is single cause and it can be addressed, it can be controlled. Not the same with cancer. There are many multiple causes, interrelation between causes, and out of that, we are concentrating on one cause, which is genetics. Now, when we say genetics,
Are you
generally,
changing slide, madam?
no, no,
Acha, okay,
I have
okay.
not. Yeah. When we say genetics, the first thing comes into mind is it is hereditary. It runs in the family, right? But that is not always what is meant. Genetics means something goes wrong with the genes which is responsible for causing cancer. So, I would like to share certain authentic sources to know the information about cancer, because in current times with WhatsApp University and Instagram University and YouTube University in place, so much of misinformation and or the incomplete information is available, which may just delude us. So it is very important to scan this QR code and know how to get updated and authentic information about what when I say global cancer burden, it has numbers in various ways. In which country, what is the rate, a higher rate of which cancer, in which continent. As you can see here, cancer incidence in 5 continents, and these volumes are published every 5 years. And these are the authentic sources. It is needless to say, it is very important to know which cancers are more, which cancers are now reducing in number to give us some hope, and various, the numbers are very important, and this is the authentic source. Now, uh, when you say cancer, again, cancer cell itself is it's known as a renegade cell. This is the title of a book, a renegade cell, by Robert Weinberg. It means rebellious. So it differs from the normal cell in various ways. As you can see on this slide, in 2000, there's some 6 characteristics, or we can say hallmarks of cancer cell, which makes it different from the normal cell. So these were then studied more, and the number of characteristics, hallmarks, increased over a period of decade, and the current or very recent is in 2022, where there are about 14 hallmarks of cancer. So this keeps on getting updated, and it is very important to know for us also that what are the cellular characteristics acquired to turn a normal cell into a cancer cell, right? Now, I would like you to see this highlighted part, this arrow, genome instability and mutations. This is one of the hallmarks of cancer. It is so important that cancer, we can definitely call it a genetic disease. Again, there are chances of confusion, that what do we mean by genetic disease? Does it mean it is hereditary? No, let us understand. There are two types of association of cancer with genetics and cancer, in which what we mean is The genome which a person is born with is playing role in its etiology, in its cancer onset. Second scenario, when cancer has already taken place and in the cancer tissue, the genetic changes take place which enable cancer cell to sustain its cancer phenotype. It is known as somatic genetics. What I have worked earlier in GCRI was about somatic genetics of leukemia, whereas currently we are concentrating on the genome a person is born with. For example, you all are familiar with Down syndrome. So that is an example of constitutional genetics. So we can definitely say cancer is a genetic disease, but genetic is not the only cause. There is interplay of complex etiology. So when we say genetics, we now understand that uh, there has to be genetic alterations, which we call as mutations. Mutation again is not the right word always, because the change may not be changing the phenotype. But anyway, let us not go into that detail. But mutations that lead to cancer, uh, mutations in the cells. So what are the let us get some quantitative idea. So finally, the third point I would like you to see that there are 10 raised to 10 occasions for a gene to get mutations. So now think about how many genes we have, about 22,000 roughly. Out of them, how many or what all genes can play a role in cancer? Now things are known, right? So considering the number of cells and the number of cells dividing over a lifetime, because every time a cell divides, the genomic material gets replicated, and that is where there is window of chances of error, which is mutation. So when this mutation takes place, not only it takes place, but it doesn't kill the cell, and the cell with mutation goes on to divide, make a clone.
So,
That is,
madam,
yes, yeah,
number of cell division is more than number of cells.
yes, sir, yes, sir. Over a lifetime, this is what is estimated.
Okay,
Yeah.
because after every cell division, cell number will increase.
No, but the old cells die out, na? That is why new cells
Okay, okay. Final.
replace
So
them.
10 to the 14 is final cell number
Yeah, yeah,
after death. Hai na? Okay,
yeah, yeah.
okay,
So to
okay.
maintain that number of cells, when the cells die out, as you know, many cells are getting replaced every day, right?
Yes, yes.
So these undergo replication, and that is how the mutation can take place. So there are these many occasions. So this is large number, right? According to this number, we would expect that everybody in the lifetime will be encountering cancer and maybe very early. But that is not the case as we see. So luckily, single mutation is not enough. And of course, there is qualitative aspect, which genes are getting mutated. So that answers that as counterintuitively, because of I mean, in spite of this probability, why cancer is so infrequent. So this is a very well worked out model of colorectal cancer, cancer of intestine, which is a very ideal example to explain what I said earlier, that cancer is a multi-step process, which means that cancer doesn't occur overnight. Before it is clinically uh, uh, detected, lot of cellular changes undergo. So here you can see, so over a period of about 10 years or more, from normal epithelium of, of intestine, carcinoma is formed after accumulation of various different mutations. So this shows that one mutation is not enough. So cancer onset is known only after person meets the doctor, right? And why anyone would go to doctor? Due to some symptoms. uh, So this shows the tumor cell population doubling. A general estimate shows that first tumor visible radiologically requires 10 raised to 8 cells in that lesion. Then clinically palpable 10 raised to 9 cells. And when this goes on increasing about 1 billion cells on 10 raised to 12 cells, then this can be responsible for the person's death. Don't take it too much in a watertight manner, but this gives us an estimate, and this is why it is important to know that cellular phenomena, cancer is a cellular phenomena, but when it increases in mass, it is that time when clinically it is detected, and then something can be done about it, starting from diagnosis, prognosis, therapy, and then monitoring after the therapy and all. So, another example, another definition of cancer is it is a clonal disease. The first point, tumor arises from a single abnormal cell. Now, if a person is already carrying a genetic mutation by birth, heritable mutation, then the newly forming cells already have one mutation. So their chances of acquiring sufficient, necessary, and sufficient types of mutations are definitely higher. So that leads to early onset of cancer. Okay. So this is known as what Mendel had used, Punnett square, right? So this Punnett square showing the interaction between genetic component and environment. Sir, slide is changing.
Yes, madam. Yes, it's
Okay,
changing. Yes.
okay. So uh, it it is also given the simile of soil and seed. There was a scientist who worked on metastasis, and he was a farmer. So basically, so he gave this simile of soil and seed, that as you can understand, that it soil as well as seed both play a role in how the harvest will come out, right? So cancer etiology is also matching with that. So it can be both genetics and environment, as you can see this one, this particular Punnett square, or genetics or environment. Here, in the first example, no genetic or no environmental role in cancer onset. What do we call it? Just like in a movie, it was said that act of God. Spontaneous is also without, not without any reason. It is due to baseline errors in DNA replication, which can be responsible for certain mutations which can start the cancer onset. But here it is purely due to the baseline errors of DNA replication. So they are known as spontaneous, where no environment or genetic component is uh, uh, prominently playing a role. And in case of the second quadrant, which where genetic component is absent and only environmental component is prominent, where microbes, all the virus causing cancer, there is a bacteria also causing cancer. If anyone knows, please do respond in chat box, which is the bacteria which is known to cause stomach cancer. Then, of course, there is a large list of chemicals, uh, chemical carcinogens, certain exposures, and lifestyle exposures. What do you mean by lifestyle? The sedentary lifestyle, lack of physical activity can be one of them, or overweight and other things. So, this is the quadrant of hope, meaning if one can avoid exposure to these, any of these, one can reduce the risk of cancer. Why are we discussing cancer after all? We are interested in knowing whether it can be under our control. Can we avoid it? So it is better to understand what causes cancer and how one can succeed in avoiding. So this is a very important part. And I will go to the next slide and then come back here. Yeah. So American Cancer Society has shared and WHO, uh, they have a body called International Agency for Research on Cancer. So they study and collect evidences from research on various chemicals. As you can see in this slide, I hope you can read the chimney smoke, vinyl chloride, benzene. All these things are so very well worked out that which type of cancer it can cause, and that is how we know that these are to be avoided, right? Now, IARC comes up with a compilation of volumes called monographs to classify the risk of the chemical. This is a very important information, and in this era of WhatsApp University, all of you, the scientific fraternity, should be aware of and visiting should be visiting this site frequently to know whether something which is in discussion, whether this can cause cancer or not. What is the authentic source? One can go to IIRC monographs and check whether the chemical or, for example, nanoparticle, for example, some paint or some other chemical which you think or you have a question whether it can be carcinogenic or not. So this is the authentic source, and the example shown here is night shift work. People do have this question in mind that people who have to work in night shift in various like nurses, factory workers, flight attendants, airplane pilots, do they have increased case risk of cancer? This can be our question, right? Now, what does it say? What is the outcome of the data mining? It belongs to group 2A. So let us understand what is this. So IARC volumes, now they are 136. Today itself I have updated the data, which is updated in on 5th July 2024 on this site. So they
Tirath Patel, please close your camera. Tirath Patel.
okay. So the group
Tirath,
1,
Tirath Patel, please close your camera. It's disturbing.
group 2A, group 2B and group 3. So this is how they are defined. Sure short carcinogens like any example comes to your mind? Like benzene, tobacco, right? These are group 1, probably and possibly. So, what is this play of words? In 1, as you can see here, probably meaning limited evidence in human and sufficient evidence in experimental animal. In case of possibly, it is like both have sufficient evidence. And then there is group 3, not classifiable as to its carcinogenicity to humans, with the number today shows 499. So, this is very important information, not directly linked to my work, but I thought I should share with you so that we know how the chemicals are classified as carcinogenic versus safe, right? Okay. So, now we come back to the area of our interest, which is these two quadrants, hereditary syndromes and uh, in which environment has almost zero role and genes have the major role. And the fourth one is interactions, interplay of both. So this is again from the NCI website, this graph. Now, when we say genetics, there are multiple genes which are studied and reported. Genetic variants. I have used the word variants and not mutation because the pathogenicity is not confirmed. So, it is genetic variant. It can affect the cancer risk, but they are not very much reported. If I ask you which gene do you know which is associated with cancer, I'm sure 99% people will think about BRCA1, BRCA2, right? Breast cancer 1, 2. So these are the mutant genes which are associated with cancer onset, breast cancer. But you'll be surprised that only 5% of breast cancer patients have mutation in BRCA gene. So what is going on with the 95% cases, right? Which means there can be other factors and other genes which are mutated and lead to breast cancer onset. So I hope this makes the scenario, complexity of the scenario very clear to you. So this graph shows frequency of alleles and relative risk. So common variants which have low penetrance, I hope you all know what is penetrance. Penetrance meaning presence of mutation and the presence of disease. When both are together, the penetrance is high. Luckily for us, there are scenarios where mutated gene person is inheriting, but there is no disease, right? So you can look it up for detail. I will not go into detail right now. So this proverbial tip of the iceberg is all we know in case of genetic variants which can be responsible for cancer. And there can be rare variants with moderate to high penetrance, and you can correlate with the relative risk. See, the relative risk is much higher in these cases. So it is the other way around, that because these have more penetrance, people having cancer, either they do not go into reproductive stage and reproductive life, and or they do not yeah, or they do not survive, and hence in the what is rare and common variant, we mean the frequency of these alleles in the population. So naturally, people having these variants will be less in the population because they have not survived or they do not give birth to progeny. That is why the variants are rare and the penetrance is high. So because penetrance is high, the variants are rare. And this is how the relative risk is calculated. What does this graph tell us? It means that there are many genetic variants playing role in cancer onset which should be studied. So this uh, pedigree chart type of a diagram shows the familial cancer. Familial meaning more than two cases of cancer in blood relatives. How many, what is the load here? About 15 to 20% of global cancer incidence is familial cancer. So these are the cases where you can expect it to be in this quadrant, right? Can you see this? Only genes and also the interaction. So so in these cases where in a family there are more than two cases in blood relatives across generation, if genetic analysis, their constitutional or germline genetic analysis is done, then the changes in which genes are altered, if we can find out, and nowadays we it is very easy to do the further thing, that which what is the function of this gene, what does it code for, and when this mutation takes place, what does it lead to, right? Whether the function is affected, which pathways are affected, and can it be potentially playing role in cancer onset. So that becomes very very important, and because this information is very less reported and more so in India, we have taken up this research, and we also want to document what is the incidence of familial cancer. See, this 15 to 20% is the authentic data available from other countries but not from India. So genetic markers, genetic changes which might be playing role in our population, predisposes our population to cancer, is something which is least reported, and that is what we want to study. We wanted to get some quantitative idea. We asked our NCDIR, and they said that the cancer registries are there, but there is no provision of collection of family history of these cancers presently, and it will be important to find. That is what they also agreed. Anyway, so there is a case of cancer, right? Now, is there any way to know that it has occurred due to some environmental exposure, or uh, is there any reason to believe that, oh, this could be due to genetic factor rather than any environmental exposure? See, it's a complex scenario, but one can solve or one can have some clue when to suspect genetic etiology on cancer in cancer. So it is not unusual to see colorectal cancer, prostate cancer in people of more than 60, 70 years of age. But if one comes across a case where the patient is of 30 years or 40 years of age, then early age diagnosis of cancer means it suggests that it could be genetic etiology. This is very rare, but yes, this does occur. Several different types of cancers in same patient. In case of kidney, lungs, breast, if both the organs, the paired organs are involved in cancer, then in the breast cancer in male, this also suggests that there is some genetic etiology. This fifth number point, as I already said, and because the constitutional genetic change is there, there could be some syndrome also, congenital syndrome. For example, Down syndrome cases are known to be predisposed to developing leukemia like phenotype. So, that connection is also indicative of genetic etiology. And the last point, certain specific ethnic groups are known to have higher risk of cancer. For example, Jews are known to have higher risk of breast cancer rather than other ethnicities. So, identification of germline, the genetic variant, is valuable to assess cancer risk in a population and to predict absolute risk in blood relatives. Now, now you know that this scenario is all the more complex because genetic variant, like BRCA gene, right? So associated with familial cancer show different expressivity in varying ethnicities. As I said, in Ashkenazi Jews, 90% of breast cancer patients will have mutation in BRCA. What is the scenario in India? Very less few reports are available. Whatever is available was very surprising. In case of South Indian patients, breast cancer patients had mutations in CHEK2 gene and MRE11 gene, but their BRCA gene was normal. So this leads us to a question that in our population, if we don't have the data of mutated gene a particular cancer, our genetics analysis, if it is followed as per the Western population, then it will not be very useful. So now we also need to understand as scientists, as people having background of some genetic studies and all, when we say genetic, we say, okay, the gene is found for this disease, right? But then finding a gene for a disease is particularly straightforward in cases like, if you have heard the name of this disease, cystic fibrosis, where it is a single gene disease. So, this is a gene, when it is mutated, it causes this phenotype, right? So, that helps in diagnosis, in genetic counseling, in finding a drug, everything. But cancer again is very complex. There is no single gene which is in case of germline mutation. There is no single gene which is responsible for cancer. So I'll not go into details of this slide, but this just shows how gene finding exercises are done in single gene disorders versus now you can see this uh, various cancer predisposing syndromes, Bloom syndrome, Werner syndrome, and other syndromes, where you can see that there are multiple genes. There is no the gene. Even BRCA, which is known as a poster boy gene, is also not the gene for any cancer, even breast cancer, right? So many genes are reported to be altered in these different cancers, which means when we plan to do genetic analysis, can we do away with selecting some 20, 80, 100 genes uh, based on what is reported, or should we go for WES? What is WES? It is whole exome sequencing, meaning all the coding genes we should be looking at, right? In clinical scenario, the targeted gene panel is something which is done routinely, but we have taken up this research, so we would like to uh, look at the entire coding genome. So whole exome sequencing we have been doing in cases where there are familial occurrences of cancer. So genetics and cancer, these are the two different scenarios. Genetics and cancer, where constitutional genetic determinants of cancer we are studying. And earlier, what I have done in leukemia cytogenetics is genetics of cancer, where the cancer cell having a signature genetic mutation is studied for clinical importance. So now cancer genes do not come from outside unless we are talking about viral etiology, right? So which type of genes can be playing role in cancer? Three broad categories one can think of. This is the person, Dr. A.G. Knudsen, who gave this idea of tumor suppressor gene in case of retinoblastoma. I will not go into detail of explaining what this slide means, but you can look it up. Two hit hypothesis is something very popular, very well worked out. So here the scenario is this: what do you mean by break fail? The cell proliferation does not stop. What is cancer? Collection of new cells. How does that take place? In two ways. Either cells do not die or cells do not stop proliferating. So a car can enter into accident by two ways: either brake fail or accelerator always on. So second category is protogen, germline oncogene activation, which can also lead to accelerator always on. So, and the third, the quality check. When the DNA genomic material gets duplicated, the mutations have to be avoided. And DNA, when it is damaged, the repair has to be done in a most uh, efficient way. So, genes playing role in DNA repair, when they are themselves defected by birth, mutated, then that leads to something called cancer predisposing syndromes. These names, XP, AT, FP, Bloom syndrome, these names you can look it up, whoever is interested, that these have by birth defective DNA repair genes. So, naturally, one can understand that it will accelerate the process of multi step carcinogenesis, which we have seen earlier, because the chances of the key genes, tumor suppressor genes, and oncogenes being altered increase a lot. So, these lead to higher chances of cancer, and they are known as cancer predisposing syndrome. So NCI gives this definition of familial cancer, and these are the differences between genetics of cancer, meaning the cells, cancer cells showing the genetic mutation, and germline variants where person may or may not get cancer, but it will definitely uh, pass on the genetic variant to the progeny. And this is why the constitutional genetic makeup of a person needs to be studied, especially when there is familial occurrence of cancer. Now, FOMO is not only for social media. Any genetic analysis or geneticist will be facing this FOMO always, because, as I said, if you go for targeted gene panel, you may be missing on many other genes which would be covered with whole exome. But now we know that there are many non-coding genes which can play non-coding genetic material which can be coding for some regulatory miRNA or siRNA all. So that also one would think that might be playing role in some gene expression. So whole genome can be better. And now uh, uh, T to T meaning telomere to telomere, meaning the complete genetic sequence. So this tells us that the human reference genome which is available is not necessarily T to T. Now, chromosome by chromosome, they are doing it and updating the information. So this field of genetics is also getting more and more comprehensive, and the long read sequencing is also the current approach, not widely used, but yes, the results are out using long read sequences, which say that, in short, what is important for us or sufficient for us to know is that many genetic variants which were not picked up by targeted gene panel or whole exome or whole genome or T2T can be picked up by long read sequencing. So, which means that you might be always having a back of the mind. It is good to have fear of missing out, so that when we come across some normal genetic analysis, we know a disclaimer that what could be left out, right? So, uh, if we go for targeted gene panel, which generally doctors order for, this 81 genes are there. This slide does not show all the genes naturally. This list did not have the gene called ppm1d. Why I mean mentioning this gene, I will show you with our example. Came across one patient. The patient contacted us, this 66-year-old lung cancer patient. Based on our announcements, he contacted us that yes, he has. You can see here so many cases of cancer in the family. So he enrolled in our study, and we carried out whole exome sequencing in the patient and as well as his normal unaffected family members. I will directly go to the results. So this is the proband. Proband meaning the first person whom we are studying for genetics. And these are the other members of the family and which changes they were found in them. Now, how to interpret the result is another long exercise which we are carrying out in our current research project, where two PhD students and one computer science faculty is also working with me. So, once we get the results of genetic analysis, what next? Next is data mining. Now, with from the available literature, we try to match our finding with whether it is reported earlier or not. Is it similar or something identical or similar? So, various other reports we have found, and this is the summary of the finding in the family. We will not go into details of this. All I want you to pay attention to is this classification. So just like carcinogen classification, group 1, group 2, 2A, 2B and all, there is a classification by American College of Medical Genetics and Genomics regarding the possible effect of a mutation. It can be pathogenic, it can be likely pathogenic, and one is uncertain significance, meaning not enough is known to call it benign and not enough is known to call it a um, pathogenic mutation. Okay, so after this result is available, thanks to bioinformatics and other in silico tools, what my students are doing is they take the sequence and put it in the bioinformatic tools and try to predict in silico virtually the structure of the protein. What is the scenario when it is normal and what is the scenario if it is mutated? One second. Hmm. So this is the information available already. This is the gene. What does it do? Where is it sitting on which chromosome? And which genetic coordinates and all that? What are its other names? And if it is mutated, what type of syndromes can be there? Of course, these mutations are not the identical what we have found, but And also, what is the reason that we should pay attention to this ppm1d, which was mentioned as unknown significance? So, our work starts after we get the genetic analysis report, and we try to understand what is its normal role. So, you can see the ppm1d controls it, it blocks these many genetic targets. It induces this target, which is responsible for p53 gene function. And if it is negatively regulating, you can match the color code, negatively regulating the p53 gene, then as we know, the p53 gene is the master guardian of the genome. So it is definitely having role in a very important target in the genome, and that is why it is important to pay attention to this gene. So this is the mutation, this is the change leading to this change in the amino acid sequence, and what all can occur due to this change is something we have found. Okay, so I have just shared one example, and I'm not going into detail of the bioinformatic analysis or in silico tools. That becomes a very different topic altogether. I am only here to highlight the importance of genetic analysis in cases where there are multiple cases of cancer in the family. So this is, of course, the incomplete data, but I'm just showing you the summary of seven different families that we have studied, and what germline mutation there was found in them, uh, and what is the ACMG guide classification for these mutations: pathogenic, lightly pathogenic, and variant of unknown significance. So this slide, I think I should skip. It will take time. You all know genetic analysis gives you the like alphabet. Bioinformatics makes sentences out of it, and data mining gives us the interpretation. And in silico tools are here, which are used by the research scholars to understand what this genetic change can be leading to in vivo in terms of the function of the protein encoded by that gene. Earlier, and ideally, this should be done in wet lab, right? By altering the gene in similar way in mouse model, for example, or other animal model, or finding people having the same genetic alteration and look for the change in the protein function and its role in pathogenesis. Now, that is not doable always, and we have the in silico tools. So, this is how the wild type, that is the normal protein, is compared with mutated protein, and this is subjected to various in silico tools to predict whether the normal function will be affected or not. So these are various tools, and bioinformatic tools help in identifying or predicting this. Okay. Okay. So I will not go into this. We have already discussed. Yeah. So American College of Medical Genetics and Genomics is gives the guideline to um, classify the genetic variants, and ClinVer is a database which which makes use of this data mining of available data to assign the pathogenicity weightage of pathogenicity to the findings. So this VUS is something which is work in progress, WIP. So it is applied, the data mining guidelines are applied to categorize the finding. Now, as you know, if something is reported very less or first time, nobody can comment on its pathogenicity, right? So, but it is important to put this finding in public domain so that people can take that into account, and it's an ongoing process that based on the reports, publications, one can draw some inference regarding the possible pathogenic effect of a genetic variant. I hope this whole exercise is clear. And now I will share with you our case reports like this ppm1d. This was published, and just ignore the below line because this paper is now already published. Second case report of BRCA2 and other germline variants. Third paper is last week accepted, which tells about some very unknown genes which are not clinically tested, but these have definitely gone into the CleanWare database, which my PhD students have learned and how to submit our findings to CleanWare. Once they verify, that will again help others to evaluate their findings, right? So now, why should we keep on reporting these case reports, right? Is it just something like a hobby, stamp collection? And especially the many people pay a lot of attention to numbers, right? How many you have got, right? How many are seen with this? But here, the scenario is n equal to 1. It can be single case, but then still it is important because this case can give us a window to evaluate the effect of this genetic change, right? So now what do we do next when we come across such genetic finding? It depending on whether it is pathogenic or even VUS. Now the person you are dealing with people, right? So the people will ask you, what is my report? What do I do? What does it mean? Many times it does not mean anything to a treating doctor. It is not clinically actionable. But then people do have query. So there comes the role of genetic counselors. So here I am sharing with you one very important useful link for genetic. Of course, you can definitely Google, but this is the team with whom we are working on DNA Day on I think on 25th April. They launched this YouTube channel on which they give the baseline information regarding genetic counseling, and one can understand when it is required and whom to contact and all that. Okay, so this again is a link to our research in Nirma University, and this is a link to a talk given earlier. Uh, is cancer a genetic disease? Because it is available on YouTube, I thought of sharing with this student fraternity mainly. Now, finally, in the last phase of my talk, I would like to give a very important takeaway, uh, because all the cancer research is done in order to avoid it, in order to treat it successfully, in order to reduce the mortality. That is most important for the wider population. For the researcher, the focus may be some interesting finding, a publication, and all that. But then what American Cancer Society has given as a very meaningful acronym, caution, is something which one should uh, remember and take away. Because in even our postal department had come up with a stamp which said that cancer can be cured if detected early. So how it can be detected early? Only if we pay attention to the clinical symptoms which are so common that nobody would think of a possibility of cancer if there is some change in bowel or bladder habit, not immediately, right? A sore that does not heal, a wound that does not heal, unusual bleeding or discharge. Any lady will just think about some hormonal cycle and all that, and not think of possibility of cancer in the first place. Uh, Thickening of lump, indigestion or difficulty in swallowing, obvious change in mole, nagging cough or change in voice. All these symptoms, if paid attention to, and if clinician is involved in the very early stage, then if it is detected early, it can be some pre-malignant condition, then there are high chances of uh, curing the cancer. Okay, so Finally, I'll just share with you what we are doing under this research project, which is funded by Nirma University under major research project. So these are the inclusion criteria. If anybody is aware of such cases, family having multiple cases of cancer, then we carry out whole exome sequencing in them. And by looking at our this announcement in social media and all, our students are contacted by the family themselves, and this is how we collect the sample. So at Nirma University, earlier Dr. Swati Manekar from computer science department was working with us, and now currently Dr. Umesh has joined our team, who will be guiding us. Now, what is the role of this computer science faculty? Let me tell you, we are outsourcing the whole exome sequencing, right, and getting the report. But the PhD students, Shristi and Jennifer, are reanalyzing the genetic data, the sequencing data, to interpret whether there are any genetic variants, mutations, and all. And that is how they do two things: one, genetic analysis; second, bioinformatics and in silico analysis also of the from genes to protein, right? So that is what is done by them. Some of the work of whole exome sequencing, when we get the some finding, we carry out Sanger sequencing, and at Gujarat Biotech Research Center GBRC lab, we have been able to do some Sanger sequencing also. And basically, we outsource to Med Genome Labs, and I'm uh, thankful to our collaborators, Nirma University mainly for the project grant, and especially Dr. Chirag Desai, who is the member of Research Advisory Committee of PhD students, and Kailash Cancer Hospital near Baroda. They have also provided us the sample access to the familial cancer cases. And last but not the least, the patients themselves and the families who have agreed to give us the samples. Okay, ignore the date, please. Sorry. Okay. Thank you so much.